The material that we're going to go over today is a little bit because I'm trying to give a tour of new code features, new capabilities. I did my best to put those new features in a structured narrative of that, made sense of, this is a possible scientific workflow that you might go through. If you start thinking this is an odd sequence of things for someone to do in an analysis, that's because a coherent analysis is not the goal so much as a demonstration of the new features. So the URL for what we are going to be following is here. So this is set up as a tutorial on the Neon website. Um, I definitely encourage you to follow along, but you also don't have to. You can also just watch me, but I think people generally get a little bit more out of it if they follow along. I am actually going to pull that onto my second screen. We will deal with that later and get myself ready. Okay, so the first part of this is going to be in R. Most of the new features that I'm going to talk about are features available in R packages. So, like usual, we are going to start by loading all those packages. So, Neon Utilities, Neon OS, and then a handful of non-neon specific packages. We are using ggplot and lubridate and dplyr and visnetwork. All right. So the first thing we are going to do, like in so many neon data uses, is we are going to load data using load by product. So bring a bunch of data into our environment. In this case, we are going to be getting soil data from the Soaproot Saddle site, which is in California, from 2018 through 2024. So kind of the framing device for what we're going to be exploring is that we are interested in soil data and how soils and microbial communities might, interact and respond to disturbance. So what we are going to bring in is data product ID DP1.10086 and site is SOAP. Start date is 2018-01 and end date is 2024-12. Because we are including 2024 data for soil properties are still provisional. So we are going to add include provisional equals true. And now our very first new feature, a very minor one on the scale of new features, but it is one that we've been asked for a lot, which is to suppress the progress bar. So to do that, you just add progress equals false. We're also going to say check size equals false. So the progress bar, uh, obviously, normally, pops across the screen saying finding files, stacking files, gives you some information about how it's going. The challenge with that is it's sort of annoying if you're in, a batch script or some sort of automated process and you've got all this, information being dumped out. The downside is that when you're running live like this, you sort of go, is anything happening? What's happening? But we've gotten to the carrot here, so we are done. We have downloaded our data. I will say by suppressing the progress bar, we've also suppressed the messages that sort of pop out as you go that are like, hey, I'm finding the files, I'm stacking the files. If the system encounters any sort of issue, it will still print a message to the console, even if you have progress equals false, because we assume that those are things you want to know. Okay, what we're going to do next is list to environment. So this is just to take all of the items in the named list of the soil object that we just downloaded and bring them all as their own object into the environment. That'll just make the next several steps easier. Okay, so next thing is the Neon OS package. So that is an R package that we developed that has a few functions. It's a much smaller package than Neon Utilities, but it has a few functions that are specifically aimed at helping folks to work with the observational data. So the first one of those that we're going to look at is a function that checks for duplicate records. So duplicate records are one of the most common data entry errors that we run into at Neon. I think, all of you who are familiar with manual data entry understand why that is. It's very easy to just, accidentally record the same data twice. It also comes up with our lab contractors. A contracted lab will realize that there's an error in an analysis, and instead of, realizing that they need to contact us to change that data, they'll just upload the updated data. And now we have two copies of the data in the system. So there is a function called remove dupes that will check for that. So we are first going to check for duplicates in the soil moisture table. So we're going to call that moisture dupes and say remove dupes from the SLS soil moisture table. The other input that remove dupes needs is the variables file. So the way that this remove dupes function works, just to explain its background a little bit, it's not just looking for fully identical records, because there are so many scenarios where you have duplicates that are not fully identical, like the example of a lab corrects data, so it's different, but it's another copy of the same data that they've already uploaded. You won't detect that if you just look for identical duplicates. So instead, every observational data table has a primary key that is identified in the variables file. And that primary key is intended to designate, this combination of data fields should indicate a unique record. So to give an example, in the mosquito data, the mosquitoes are sorted into groups by species and sex. And so the unique combination of the original trap ID, species, and sex should always get you a single record. Because basically, if you have multiples of that, that means they made, two groups of the same species and sex within a sample, and that should not happen. So that's why the variables file is included in the inputs to remove dupes, is to get that primary key. We run that, and hey, no duplicates. Very exciting. We are now going to do the same thing for the soil chemistry table. So remove dupes from the soil chemistry table, the same variables file, obviously, because it's the same data product. That also has no duplicates. So this gets at another aspect of this. I had a fun time trying to set this up for you and trying to find, a relatively recent and interesting data set that had duplicates, and eventually I gave up. So what's happening is we also use these code tools internally. So the Neon Science staff are using these functions to check for duplicates in the data and then to fix those duplicates. And so uh, in the last few years, as we've been using these new features, the number of duplicates in the published data has absolutely gone down. And so you will see this less. But I do still recommend checking for duplicates, particularly if you're using provisional data. So that's, the most recently published data. Maybe we haven't had time to get in and fix those data yet. The provisional data are more likely to have duplicates at this point. Okay, so this example does not have any duplicates. So what we're going to do now is we are going to join the chemistry data and the moisture data. So
We got one
yeah,
question in the chat.
I have a question in the chat. Yes. Okay, yes. What is returned? There's actually an extra column added that has, I think it's called duplicate record QF. So a quality flag for the duplicate records that will, okay, so there's an additional element in the remove dupes function, which is that it also tries to resolve the duplicates if it can. It's based on some fairly simple criteria, so there are a lot of duplicates that it can't resolve. But like for example, if it finds that, actually these records are entirely identical other than their unique identifier, it will just merge them into one and say, okay, like those were duplicates, but they were identical. We're going to assume they're the same thing and they merge into one. In that case, they get a flag of one. The other option is if it can't resolve them, it flags them with a two and just leaves both records in place. I am actually planning to make an update to the package that would have a flag only option that just doesn't even try to resolve, that just flags these are the duplicated records. But yeah, the idea really is just to some extent, what to do with the duplicates is your decision as a user trying to figure out what to do with an analysis. And so we've tried to take a fairly conservative approach of just here are the duplicates. We only remove the obvious ones, see what to do next. Thanks, Dave, for alerting me to the question. Anything else about that? Okay, great. Okay, so we're going to join the tables now because we are going to take a look at whether soil chemistry varies in some predictable way with soil moisture. Thinking about sort of workflow, you're always going to want to check for duplicates before you join, because if you think about joining tables and maybe each of them has duplicates, they potentially start multiplying with each other and you get, a bunch of extra rows that you could just take care of if you remove the duplicates first. So now we join the tables and we do that using another function from the Neon OS package, which is join table neon. So this function was highly requested. I think a lot of you have worked with observational data. It comes to you in many tables, and often you want the data in those different tables together. And so having a standardized automated system that knows what the two tables join on is super helpful. So we are joining soil moisture with soil chemistry, and there it goes. To show you what that function is using as its basis for doing that, I'm just going to jump out of R for a second and go to, this is the data products details page for the soil properties data product. So we're on the Neon data portal now, and I'm just going to scroll down past some of this to, this is like a little sort of documentation reader on the details pages. And the default that shows up in this reader is always the quick start guide. And the quick start guide, there's the top of it, but I'm going to jump down a few pages. The quick start guide for observational data products is always going to include this table joining table. So this table in the quick start guide tells you for each pair of tables in a data product, what is the variable that they join on. It does in some cases say, this is not a simple, just join on these fields. This one is quite long because there are many tables in this data product, but soil chemistry and soil moisture are here at the bottom, and you can see they join on sample ID. So obviously you can, use this information from the quick start guide, join tables yourself. Join table neon is just pulling from this data set to do that little bit of extra work for you. Okay, so now we have we have data in one table and we can check out whether there's a correlation between soil nitrogen and soil moisture. Again, not necessarily a compelling scientific reason to ask this question, just something to liven up the tutorial with some figures. So we do see at least somewhat of a correlation where the soil plots that are wetter also tend to have a somewhat higher nitrogen. Okay, so I said up top that sort of the premise for what we're doing here is that we're interested in, oh yes, what happens in join table neon function if you try to join two tables that don't have an easy join. It should give you a message that basically says, I don't know how to join these directly. Go and check out the information in the quick start guide. Um, so it should pretty reliably do that. And tell you, where to go and look. There is, coming back for a second, to sort of the Neon OS package in general, there is an existing tutorial for the Neon OS package that goes into a little bit more detail on some of this, and it also gives an example. One of the most common types of I can't join this easily is if, say, you have three tables and one of them is effectively an intermediate for the other two. So you can join them in series, but you can't actually join number one and number three directly. So that tutorial walks through how to do that triple join if that's the scenario. And that tutorial is linked from the tutorial that we're going through now. So yeah, definitely check that out. Okay, so I said up top that sort of the framing device for exploring these soil data is that we're interested in soil chemistry and soil microbes and responses to disturbance. So, if you're interested in disturbances at neon sites, the site management and event reporting data product is going to be of interest to you. So that data product records things that happen at the site. Um, so it's very broad. I think that is one of the challenges with it because, it could be controlled burn, it could be a wildfire, it could be, this is a managed site and they moved the cows in. It could be this is not a managed site, but we had some sort of like wildlife event. The cows got out and came into the site. So it's really just the field staff trying to record anything that happens in the vicinity of a neon site that we would expect to impact the sampling, but that is not, explicitly part of neon sampling. This data product, we're aware people have found it challenging. And so there is a new function in Neon Utilities that lets you search it a little bit more easily. So what it does, you can still download site management and event reporting like you would download any other data product. And what you're going to get is just the record of all of the events for the sites and the time periods that you've chosen. What this new function does is it lets you look for specific types of events. So what we're looking for in this particular tutorial is fire. So Soaproot Saddle is in California. We know there have been a lot of fires in California. Let's go and find out, have there been fires at Soaproot? So we are going to call this object sim. That is just the abbreviation we use for the site management data. And we are going to use the function by event sim to search for events labeled as fire at the site soap. So this will take a minute because it has to, go search through all the soap data. It takes a little bit of time, but it is faster than downloading all of the site management data from soap and then going through it after the fact. Okay, so we have done that search, and now just to show you what that looks like, you're going to get the same output from this function that you normally get from load by product. Basically, you're going to get, the data table, the variables, the metadata tables. It's really just that this event data table is going to be subset to the specific type of event that you asked about. So actually, I'm going to use view to look at that more easily. So look at what we downloaded. We've got just 6 records. So we can see event type is fire. That's what we searched for. And then usually for these types of events, there's going to be a little bit more information as you keep looking through. As you can imagine, because different kinds of events are captured here, there is a little bit of manual work involved in just, checking out what data you've got. You can see the first event that was recorded was a controlled burn. The other 5 records are all wildfire. Uh, for the wildfire, fire severity was rated high. And if we scroll all the way over to the remarks, you can see they've noted sort of which fire we're talking about. So controlled burn is just a controlled burn. And then there's these 4 records about the creek fire, and the final record is about the blue fire. And if we scroll back to the beginning, we've got start dates and end dates. The creek fire was in 2020 and the blue fire was in 2021. So, a relatively efficient way to get information about fires at this site. Okay, so now the question is, if we're interested in, the impact that this fire may have had on soil chemistry and microbes, what kind of measurement dates do we have for the chemistry data? So to get that, we are going to look at the years represented. I see there's a question in the chat, just a second. The years represented in the soil chemistry collect dates, 2018 and 2024. Okay. Ooh, yes. Can you see which plot IDs were affected? Yes. We're actually going to get to that in a minute, but I can show you now. There is this location ID field. Oops. I thought there was a way I could make that expand. Maybe like that. Yeah. And so this location ID field is just a comma separated list of all of the locations that the field staff evaluated as like this location was affected by this fire. I think for the creek fire, I was looking at this earlier, they list many, many plots, and then at the end they just list soap. Like the soap site was affected by this fire. But, it depends a little bit on the specific incident, but what this represents is sort of the field scientist's best estimates for these are the plots that were impacted by this event. Okay, so we have soil chemistry data from 2018 and 2024. And the fires that we just saw in the event data happened in 2020 and 2021. So the fires did happen in between our chemistry data. The chemistry data is not necessarily, right adjacent to the fire, but, we would theoretically see impacts from the fire between those dates. So just again, to keep things interesting, we are going to look at that same relationship between percent nitrogen and soil moisture for the 2018 and the 2024 data. Obviously, other things could have happened in those years besides a couple of fires, but we can see if there's, a difference in those data or that relationship between those dates. So we are again going to compare percent nitrogen to soil moisture. This time, the data is going to be first. We're going to look just at the data from 2018. All right, so that is just 2018, and then add in the points for the data from 2024. Oh, and I need to change the color. All right. So, maybe not an obvious difference in pattern between those two dates. Again, we're just looking at nitrogen and soil moisture. There are many other things that, people could look into. In particular, if you start looking at which plots burned and you go, oh, actually, some of these points represent plots that burned and some of these points represent plots that didn't burn, you might be able to start separating out more details. Just a possibility, and we're not going to get into that today, but something to think about in terms of the other data that's available here. And that is also a sort of stay tuned feature. We are actively working on a code package specifically to work with the location data in the site management data product. We had an intern this past summer who sort of got that project started, and we are working on wrapping it up to just give you better tools for extracting this location data from the site management data product and, using it for whatever you need to use it for. Okay. Next up, I also said at the beginning that for this hypothetical data analysis, we are interested in microbial data. So this next sort of new feature may not be, well, some of you may not have been aware that this was an issue before, but we have data on microbial taxonomic identifications. And until fairly recently, those data, it was a sort of a iterative code process to get those data and get them stacked into a single data table. They weren't included in what got stacked by Neon Utilities. Without going into too many details, it's sort of because those files were too big, but that situation has improved. And so you can now get the microbial taxonomic data also stacked through load by product. So what we're going to do next is get the microbial taxonomic data also from a soap root saddle from this same time period. So to do that, we again use load by product. Data product ID in this case is DP1.10081.002. This is one of the very exciting data products that is on its second revision. There are only a handful of those, so for the most part, this is still 001 for nearly all data products, but we do have a few 002s now. Site is soap root. Start date 2018-05, and end date is 2024-12. In this case, we need to get the expanded package because those taxonomic identification files are in the expanded package. We are going to include provisional. I am again going to turn off the progress bar just to remind you all that that exists and skip the size checking. So this is also going to take a little time. Like I mentioned, those microbial taxonomic identification files are really big, and so it takes a little bit of time to do the download and the stacking. Both of those steps are a little bit slow for these data. While we're waiting for that to happen, any questions? Okay. Computer is still thinking. And again, without the progress bar, we don't know where it is in that process.
Claire, I could add about that SIM data. You
Yeah.
did a great job describing the package that we're working on. And it really depends, and we're trying to get better at this to the extent we realize that the exact plots that are impacted are really the data of interest in most cases of the disturbance, because those, particularly with the observational data, that those disturbances, depending on the extent of and the severity of them, could really impact the data. So it's kind of, unfortunately, it's a little bit variable. Sometimes we're really dependent. Fire is a little bit more clear. Sometimes we're dependent on a site host to understand which plots have been treated with respect to something like invasive species, or if it's an agricultural site, some of the timing of crops and some of the sprays that were put on them, et cetera. So the data are a little bit variable, unfortunately, depending on where we get it from, and also to some extent the resources that our field crews can dedicate towards collecting it. So can they visit each plot after a fire and make a specific determination, or are they creating that based on a fire map or something like that?
Yeah, thanks, Dave. That's a great point. That yeah, the that location data is as best as we can get it. But it's there are definitely some challenges to knowing for sure exactly which plots were affected. Okay, so our download has completed. You'll notice that you do get a little alert message here. Um, this is really just, again, like I said, these files are really big. And so if you try to download a really large amount of data, there's a fairly high risk that, something will happen. Your laptop won't quite be up to the job just because of the processing power of a laptop. And so that warning message just pops up to say, hey, if you're having problems, try downloading a smaller amount of data. And we're going to do the same thing we did before, pulling the individual tables directly into the environment. And now we can take a look at what are the fungal taxonomic units that were found in these data. So we're going to use ggplot for this one, and we are going to plot the per sample taxonomy for ITS. So ITS is the gene or the gene region that we use for sequencing for fungi, and we are going to plot by phylum within that. So, going to make a stacked bar chart. And let's see, this is just making the axis slightly prettier since there are going to be taxonomic names along it. Okay, element text. We want to angle away from the axis. Okay. And then boom. Okay. Well, that's very sad. When I have done this before, you actually can see gradations of gray in these bars to see, sort of the numbers of finer taxonomic units in there. I don't know if that is going to show up if I make it bigger. Not really. So trust me that there are, in fact, finer gradations of taxonomy within those bars. So that, again, is just, a plot to demonstrate that we have, in fact, got these data. We've been looking again at this disturbance, and so trying to assess whether we have different results at different time points. So we want to check out what are the years that we have within this data that we downloaded, because we downloaded from 2018 to 2024, but what did we actually get? Only 2018. So what's happening there is they did sequence marker genes at SOAPROOT in 2024. We just don't have the taxonomic analysis back yet. And so that is data we expect to have in the near future, but it's not available yet. So getting at this question, let me think about how much time I have left. Okay. So this question of, you searched for 7 years of data, but actually only one of those years had data. And are there ways to take a look at, we have all these soil samples, which of those soil samples were actually sampled for microbes? If we have time, I'm going to come back to this at the end. There is a function in Neon OS for looking through sample hierarchies. But for now, I'm going to skip ahead and talk first about the neon plants package. So we were taking a look at soil chemistry and microbial communities. We might also be interested in the relationship between soil chemistry and plant communities. We are very, very close to releasing a package focused on working with neon plant data. And that package is going to include both community, data applications and things like biomass and productivity. It is not quite out yet, but stay tuned. That will be coming very soon. So now I am going to jump to talk just a little bit about Python because we've been talking about the Neon Utilities package, and that is what most people are familiar with. And if you're happy working in R, fantastic. That is where we are. But we do have now a Python version of the package. And so I'm just going to demo that quickly and make sure that this is actually, yeah, no, Cyverse has logged me out. That is why that happened. So this is a Python Jupyter notebook in Cyverse. You will see in a minute why I'm doing this on Cyverse instead of on my local machine, but don't worry, the Python Neon Utilities package will also work locally. This is just so that I can demo another feature in a minute. Um, so if you've been following along in R, don't worry. We're just going to do a couple things here, so don't worry that you have to immediately jump to Python and do things there as well. So we are going to import the Neon Utilities package. Great. Typically, the way that I do this is to import it as new, just to not have to type out the full package name. But you know, you do you, whatever works. Um, and I'm just going to demo that basically the Neon Utilities package in Python works exactly like the Neon Utilities package in R. So we have a load by product function that takes the same inputs. So I'm just downloading the same soil data that we downloaded at the beginning of this exercise. So data product ID, site is soaproot, start date 2018-01, and end date 2024-12. Again, we have the include provisional option, and I'm not going to suppress the progress bar this time because I think the progress bar looks kind of nifty in Python, but you can do that. And this should, oop, nope, I do that all the time because I'm too used to R. I forget to specify the package at the front. Okay, so while it's downloading, just to highlight the sort of small ways that this is different, you can see the sort of syntax and formatting is very slightly different. We call this snake case instead of camel case. So with underscores in the function names rather than, using lowercase and capital letters to indicate different words. Include provisional and check size here. Also use underscores instead of dots. Dots have like programmatic meaning in Python, and so you don't want to use, well, you can't use dots in things like function names and inputs. Um, but otherwise, it's the same inputs, the same process, and what you get back is going to be, again, different because it's in Python than R, but fundamentally the same thing. So in R, you get a named list of data tables and metadata tables. In Python, you get a dictionary, and the dictionary has, named tables and metadata tables. They're going to have the same names, they're going to be the same tables. We have tried to make it as as similar as possible. Where there are a few differences, we have not brought over all of the functionality of Neon utilities into Python yet. So the sort of basic download and stack functions, load by product, the AOP download functions, the independent stacking functions, those are all there. We haven't brought over stack eddy yet. So that is the function in R that stacks the data from the surface atmosphere exchange data product. We are very, very close. We have a Python version of that function drafted. It just hasn't been sort of fully tested and finalized and added to the published package. So I am actually hoping to get that out in the next few weeks. The other thing that we don't have in Python yet, we haven't added the sort of other R packages. So Neon OS and GeoNeon, we do not have in Python yet. So I think this is just, a question of having the time. And, the goal certainly is to have a full suite of functionality in Python as well as R. It just may take us a little more time to get there, but definitely feel free to reach out to us if there's particular things that you want to be able to do in Python that aren't supported yet. Okay, the next thing that I want to demo, and this is why I am in Cyverse for this part, is a new feature that has to do with the backend load by product. So what load by product is doing behind the scenes is downloading all of the data files to a temporary directory on your machine and then stacking them into the particular data tables and bringing those into the code environment. Um, but the world is producing all kinds of new and exciting cloud native software tools. Um, it's like just a very, very active area of development right now. And so one of the things that Neon is doing that I think is really exciting, but that we are still kind of figuring out, is figuring out how to best take advantage of those tools. But one thing that a lot of those tools can do is basically let you skip the download step in what I just described. You point it to all of the data files out there in the cloud, and it grabs them all and brings them in and stacks them into data files in one step. Um, and so we have added an option to load by product called cloud mode. And if you are in a cloud environment, you can use that feature to go and grab the files from the Neon cloud, bring them into your cloud environment without doing the intermediate download step. And it is much faster. So I'm just going to copy that same load by product command, and I'm just going to add cloud mode equals true, and off it goes. And you'll see it went straight to stacking because it skipped the finding and the downloading. Technically, it did do the finding, it just did the finding real fast. And so I didn't bother including a progress bar for that step in this scenario. But it stacked the data files, and here we are with the same data. Hooray! So that is intended, like I said, for using when you're working in a cloud environment. That feature, cloud mode equals true, is also available in R. So that will work in either language and does the same thing in terms of using those cloud native tools. Thanks, Ty. I love it too. It's very exciting.
Oh, that's a game changer right there. That
Yeah.
that opens up, especially with the AOP data and stuff. That's amazing. That's a really big deal. Is that
Exactly.
not no AOP data yet for that cloud mode?
No,
Well, either way, I'll still appreciate it for all the other, but it just yeah, that makes it a lot easier. Appreciate it.
yeah. Yeah, unfortunately, this is strictly for tabular data files. And so we are exploring cloud native options for the AOP data. I think that is going to be there have to be some changes on the back end before we can make that work, but we are definitely looking into that.
Sweet. Thank you.
Yeah. Okay. I think I do have time to jump back and just talk a little bit about querying samples. So I'm going to go back to, so this is, like I said before, basically trying to help you with questions around like, okay, they collected a soil sample in the field. What else did they do with that sample? Like, I see there's some soil moisture data, there's genetic data, but there's only genetic data for some of the samples, and sort of, well, exactly which soil core did those genetic data come out of? Questions like that, or potentially going backwards if you're working at the biorepository and you get a soil sample out of the biorepository saying, hey, what is the original core that that came from? And can I find the data associated with that original core that this archived sample came from? So the Neon OS package has a function called get sample tree that finds the sample hierarchy for the sample that you ask about. So what we're going to do is run get sample tree on just the first sample in the soil core collection data that we downloaded. So I'm just saying there's a list of sample IDs in the soil core collection table. Give me the first one and find its sample tree. So this is going to take a second. Oh, actually, that was pretty fast. And what you can do with that, I'll just briefly show you what that looks like. It's a bit esoteric. You're going to have a big list of sample tags, barcodes, and parent sample tags and barcodes. And so this is all of the samples that are, you can think of it as like descended from that particular soil core. In order to get that sort of out of that spreadsheet view and something that's a little bit easier to visualize in a little bit more of a tangible way, I like the Viz network package. It's just a package for creating data graphs, so directed graphs. And so we're going to, the way that it works is based on edges and nodes, which is basically, you can think of the samples are the nodes and the edges are the pointers between the samples. So we are going to, so what we're saying here in defining the edges, all right, we have 5 minutes left. That is enough time to show you this fun graph. So this is the from and to, this is defining the relationships. So basically saying for each sample and its unique identifier, where it comes from is the parent sample. Um, that's how that is defined. And then the nodes are just the samples themselves. So the sample UUID, and then we are going to label them with the sample tag. So I didn't actually run that one. And now we can use that to make a Viz network graph from the nodes and the edges I'm going to draw arrows on it. All right. So this is fun because what you have now is this image of this here is the original parent. And you can see from the arrows, it created a whole, or we created from it a whole bunch of subsamples. One of those subsamples, this one, then went on to create another subsample that then created 5 more subsamples. 6 more, I miscounted. And so this can be really helpful for sort of understanding the sample workflow. Question in the chat, can you only get descendants? No. Basically, if you ran this get sample tree function on any of the samples in this set, you would get this same result. It's basically, it's taking a sample and looking both up and down the hierarchy as far as it can go in every direction, and then returning the aggregate. So basically, these are all the samples that are directly related to whatever sample you started with. I will say, in terms of sort of use cases for this, because you only query one sample at a time and get, one sample family at a time, It's not something I really expect people to use analytically. It's a little too narrow for that. But particularly if you're looking at, a relatively complex data product where, a parent sample is then used to do many different things like this, it can be really helpful to just understand those relationships and understand, okay, here's where I started. And, I can see data associated with each of these other samples. Here's how they're related to each other. Here's how, one sample got turned into another. And like I said, also related to Jess's question, because you can go in either direction, you can start sort of anywhere on the tree. If you start with an archived sample at the biorepository, it can be very useful for then going back up the tree and seeing what the samples higher in the hierarchy look like for that particular sample. So I think it's a nice sort of exploration and understanding tool, even if it's not quite as much of an analytical tool.
So could I use this
Yeah.
with the biorepository data where it's like, okay, I have the plot and then I have the shrew and then I have the two ticks. Will this show me the network of that relationship now?
Did the ticks come off of the shrew, or are the ticks
Yeah,
just from that plot?
the ticks off the shrew and the shrew off the plot. I
I think this should show you that relationship. That depends on exactly how we've designated the ticks and like if the ticks are subsamples of the shrew.
right.